Hi there, fellow primates. I hope this video finds you well on this alternatingly sunny and overcast Friday afternoon, where sometimes snow falls in London in April. So welcome to our crazy world. You may have noticed that I haven't recorded videos for a couple of weeks because I have been traveling for the first time in more than 2 years. I jumped on a plane and flew somewhere else. I flew to Norway this time. I was invited to give a seminar at the Center for Ecological and Evolutionary Synthesis at the University of Oslo. And that was just wonderful because it's been so long that I've been in a room with other people live there to exchange thoughts and to present my thinking. In this case, they invited me to talk about my forthcoming book, which I'd not done before. So it was an interesting experiment to see if the ideas that I have were received well, and there was lively discussion afterwards. So that was all very successful and very enjoyable. And I stayed for a week because I also visited my friend and colleague Ivan Undheim, who works at the University of Oslo, to catch up, have some beers, which in Norway are offensively expensive. A month ago or so, I was in a pub in South Kensington here in London, which admittedly is an expensive part of the city. And I ordered a pint, and they gave me the pint and said, "Sir, that is £7.45. And I was like, alrighty then, that's a lot of money. But for that amount of money, you wouldn't be able to get a pint in most pubs in Norway. Nevertheless, we did lubricate with beer because it is so pleasurable to do that. We spent a lot of time outdoors as well. It was wonderfully crisp, sunny weather when I was in Norway, so I went out walking a lot with Ivan and his dog Troll, which you can see here, which is a German wire-haired pointer. An amazingly cool dog that he's training, so it can be a gun dog, so you can go bird hunting with it. And I fell in love with the dog, I guess, as any person would. I am actually a lover of small dogs. My favorite breed probably, well, one of them anyway, is the Chihuahua, which when I tell people this, mostly triggers laughter and finger pointing. But this big, highly energetic dog also, awoke something in me. So perhaps at some point in my life I should buy a dog as well. Although it's hard work to train them, I've noticed. We also did some barbecuing. This is Ivan bent over his chest freezer in his garage, where he stores more than 100 kilograms of meat. So I felt a little bit like a kid in a candy store. Oh, what sausages and what chops are we going to eat tonight? But it wasn't all fun and play. We also worked, which is fun and play as well. We even behaved like real scientists. We got the whiteboard out and we drew up a list of all the data that we have generated for centipede venoms, transcriptomic data, proteomic data, to figure out what else we're going to do with these data sets and the stories we would like to tell with them. Anyway, very, very nice trip. But that's not why you're tuning in. It's time for Journal Club. And this week's Journal Club is about reloading weapons. One of the most neglected subjects in movies, for instance. So if you watch a movie, I don't know, Rambo or John Wick, what you see is the people firing their guns. You see bullets flying and hitting the targets, foreheads caving in, limbs shattering, because that's where the visual excitement is. The use of ammunition flying through the air, bullets landing on target, that's where the excitement is. What you don't see is close-ups of the type of ammunition that are put into guns. And what you see even less is a John Wick or a Rambo carefully putting bullets in their revolver or in the magazine of a machine gun. You don't see that because that is boring television. It's not interesting to see how people reload their guns. And the same is true in the world of Venom. If you watch a popular show on venomous animals or you read a popular science article about venomous creatures, what you see is the action. You see the snake bite, you see the wasp or the scorpion sting, you see spitting cobra spit. You can see the ammunition flying through the air. And if you watch an R-rated program, you may even see what happens to the victim. You may see the bloody limb of a person bitten by a rattlesnake or a twitching rabbit that has been bitten by a venomous snake. What you see less of is the ammunition itself. If you want to see venom ammunition, you have to consult the scientific literature, and their scientists dutifully present their little pie charts in which different slices of the pie chart show the components of the major venom toxins. What you see even less is literature about how venom glands are reloaded once they are depleted. Very small literature actually, with only a few articles, mostly on snakes, scorpions and centipedes, that address the question, how do venom glands replete themselves, replenish themselves, regenerate themselves? In part, well, because the major findings that have been discovered so far are obvious, predictable and boring. 1. Reloading a venom gland takes time. How much? Depends entirely on circumstances or what species you talk about. Certain species of parasitoid wasps, tiny little venomous creatures, can reload their venom glands in a matter of hours. Other venomous creatures like spiders may take days or even a bunch of weeks before the venom glands are fully replenished. Second finding that is unsurprising and exciting is, well, if a scorpion, for instance, is replenishing its venom gland, it uses more energy, it uses more oxygen than a resting scorpion. Well, that's not exciting, but it's good to know. But there is one area of the study of reloading venom glands that is sort of exciting. And that is the question addressed in a paper that was published in this month's issue, the April issue of Toxicon, titled Contrasting Patterns of Venom Regeneration in a Centipede and a Scorpion. And this is written by a team of biologists at Florida State University. The leader of this team is Darren Rokita, who's been leading an excellent venom research team with different makeups over the years. And the first author, Gunnar Nyström, is one of the PhD students that led this research. And what they did is they looked at 2 venomous species they could collect in the wild in Florida. A scorpion, Centruroides hensai, and the giant blue Florida centipede, Scolopendra viridis. And they got 16 specimens of the centipede, all males, and 15 female scorpions. And the question they wanted to address is, well, the venom is composed of a number of different chemical bullets, different toxin families. When you have a depleted venom gland, do these toxin families all replenish at the same time, at the same rate? Or do certain venom components get refilled quicker than others? So what they did is they generated reverse phase high performance liquid chromatography profiles for the venoms of these species, which you can see here. Centipedes at the top, scorpions at the bottom. And you can see these peaks. Now, and although they didn't do sequence analysis to find out what all these peaks represent, the specific toxin families, we know from many years of research across venomous creatures that these peaks represent distinct major components in the venom. So for the centipede, let's just focus on the centipedes in this paper. They created 13 clusters of major components based on these peaks in the chromatogram. So 13 peak clusters. And they wanted to know, if you have an empty venom gland in the centipede, do all these clusters replenish themselves at the same rate? So they took the 16 specimens of the centipedes and they divided them into 5 experimental groups. One that was allowed after the venom was depleted to replenish for 1 day, another group replenished for 2 days, another 4 days, another 10 days, and another 2 weeks. And what you see here in this graph is, if you look at the protein content of the replenished venom, you can see the leftmost box plot shows the protein content of the initial venom glands, and then the next 5 to the right of it, the protein content after 1 day of replenishment, 2 days, 4 days, 10 days, and finally on the far right, 2 weeks. And one conclusion you can draw from this graph is that in Scolopendra viridis, this Florida giant blue centipede, it takes at least 2 weeks for the venom gland to fully replenish itself, the protein content to be on the same level as it initially was. So that's interesting. But more interesting than that is that indeed there is an asynchrony in which components replenish. So over the time course, from 1 day of replenishing to 2 weeks of replenishing, different peak clusters, different sets of major venom components replenish quicker than others. So there's an asymmetry in the abundance of different venom components at different times of replenishment. So it's not all at the same rate. Now, is that interesting? Yeah, that is interesting, because if you have different abundances of venom components replenishing at different or different abundances at different times during replenishment of a venom gland, that means that perhaps the function of the venom gland can differ at times. The animal may be able to tackle different types of prey only after a certain time, when certain venom components have a certain abundance. That's just speculation. The interesting thing, though, is that they find, although there is an asynchrony in protein abundance across regeneration time in the centipedes, if you look even after one single day, in the experimental groups, which contain mostly 3 specimens for the different regeneration periods, except for the 2 weeks, that was 4 specimens, at least 2 of those 3 or 4 specimens after one day contain at least detectable peaks in all 13 peak clusters. In other words, even after just a single day of venom gland replenishment, when the venom gland is by no means full at all, you can already detect all major venom components. Now, why is this interesting? Well, it's interesting if you want to do bioprospecting, for instance, and you're not interested in the different abundances of venom components, you just want to know what is in the venom. You could, for instance, collect some centipedes in the wild, a species that's not been studied before, and you milk the venom and you wait 2 weeks until the venom gland is full, or a week or whatever, and then milk the venom again until you have a lot of it, so you can convince yourself, you at least then catch everything. This experiment shows that at least for Scolopendra vividus, after one day, you already will find all venom components present if you have a sufficient number of specimens. So you can go into the wild, catch an animal, milk the venom of a number of specimens, release them immediately. You don't have to keep them in the lab for a week or 2 weeks or longer, because already after only a day of replenishing venom glands, you will be able to find all the major venom components that are present in a fully replete venom gland. Now, in contrast, they found that in scorpions, the relative abundances of the different venom components do not change over time as the venom gland replenishes, so that's a distinct difference. And those are 2 of the major findings of this paper. It is a very elegant paper. They did a lot of detailed statistics, principal component analysis. We shall not go into details to find out these things. Now, why is this interesting? Other than, it will allow people to inform how they milk centipede venoms, which I will end this video with. I will show you some photographs of how you actually milk venom in a centipede. And there are a few reasons why this is interesting. As I alluded to, there may be functional consequences if you have asynchronous venom replenishment. So a paper that was published, I think, in 2019 by Edson, Norberto, Carcamo, Noriega and colleagues. They studied a different scorpion in the same genus, Centruroides limpidus, which is a Mexican species, and they found that there is, in contrast to Centruroides hensi, asynchronous replenishment of different venom components, and that has a functional relevance because the toxins that are used to subdue prey, in the case they tested this on crickets, replenish fully by 10 days after depletion of the venom glands, but the toxins used to deter predators, pain-inducing peptides, only replenish on day 13. So they showed that for this species of centipede, scorpion, the toxins involved in prey subduction or in prey capture replenish quicker than the ones used for predator deterrence. I will be back in a sec because I will explain in a moment. Alright, I'm back. Sorry, I had to run downstairs because it sounded like somebody was centrifuging a ton of bricks. We had a new washing machine installed yesterday and we did one load of washing and it made a god-awful noise when it was spinning off. So today we did another load of washing on a different program, carefully measuring the amount of laundry we put in there, which is not even half of the capacity it can take. And again, when it spins off, it is like the house is going to fall down. So there's something faulty. I'll have to call the installers again after I finish recording this. Anyway, that was distracting, all that noise in the background. So where was I? Yes, the asynchronous replenishment of venom components used for subduing prey and deterring predators in a scorpion. Another paper that was published a few years ago by Zia Nisani et al. Looked at a different species of scorpion, in this case a scorpion from South Africa, and they found yet a different story to do with asynchrony of replenishment of venom. As you can see in this image, this particular species of scorpion has two types of venom. It has a clear looking pre-venom and a milky looking normal venom. And it turns out that the pre-venom has a high concentration of potassium ions, which are pain-inducing in mammals, and a mixture of peptides. And the milky venom that comes out after the pre-venom has been expended is fuller of a more complex cocktail of proteins. And it turns out if you deplete the venom gland of this creature, the pre-venom replenishes quicker. And the pre-venom is very good for subduing prey, but also very good for deterring predators, because that is the metabolically cheap potassium ions that induce pain and therefore deter predators. So in this scorpion species, the ability to subdue prey with the replenishing venom and deter predators replenishes at the same speed. So there's an interesting story there about the functional connotations of whether venom components replenish synchronously or asynchronously. Probably has something else to do with something else as well. If you look, for instance, at the centipede venom glands, data suggests that all the different venom components are not homogeneously distributed throughout the venom gland. There's a heterogeneous distribution of different toxins, and hence where they are produced in the venom gland. And if that is the case, then perhaps the replenishing activities of different parts of the venom gland are different from those in other parts of the venom gland, and that may be reflected in an asynchronous pattern of toxin replenishment. So this is an interesting topic, but as interesting as it gets. So I won't, in this series of journal clubs on video, come back to this topic very often. But since it is an integral part of how venom functions, the reloading of the venom weapon, I thought I should at least dedicate one short video to it. And so I have done that now. I did mention a little bonus. If you've never worked with venomous creatures, and you may be very curious, how do you actually get venom out of the various creatures? Well, it depends on the creatures, but since I work on centipedes, and the Nystrom et al. Paper looked at centipedes as well, how do you get venom out of them? The first time I learned this was when I met Eivind Undheim at a conference a number of years ago in America, and he had a centipede in his hotel room, and he had his milking equipment, which is essentially an electrified forceps, and he showed me how to do it. So when Eivind was visiting me before the pandemic, I think just before the pandemic, with a master's student, he had ordered in something like 20 giant Vietnamese centipedes, one of which you can see here. It's been anesthetized and immobilized with rubber bands on my AeroPress coffee machine, and that's an excellent piece of equipment if you want to milk centipedes, because it has a round tube where you can easily stick the centipede on, and it has a rim at the top with straight edges. And so, as you can see in this photograph, you can lay it on its side, and the straight edges make sure that it's stable and angled towards you, so you can look under your dissection microscope and actually milk the venom by, as you can see in this picture, applying a electrified forceps to the base of the venom claws, putting a small current to it. A droplet of venom will form on the tip of the claw, and then you can use a pipette tip to suck it up, and ta-da, you have your venom. Now, in theory, this is far easier than in practice, because these creatures, you want them to wake up first, because the muscles surrounding the venom glands that need to contract in order to expunge the venom, if you use CO2 as we do to anesthetize them, they get paralyzed, they don't work well. So you need to wait until the animal wakes up, hence you need to immobilize it with rubber bands, because only then do you get a nice venom yield. But as you can see, and let's go back to this image, the posterior legs of this animal hang off the edge of the AeroPress, and which means if it wakes up, it will start whipping the hind part of the body through the air to try to find purges. And if it catches you, for instance, on your sleeve or on your glove or your hand, their claws are extremely sharp, and they won't let go. Some species can actually, puncture your skin easily with these with these very sharp, pointy legs. So what we then did is we had a little cardboard box in which we sort of inserted the dangling hind bit of the body, and then you could hear it stretch around when it woke up, which was a good sign. We then knew it was awake and we could milk its venom. So that is how you milk venom from centipedes. That's the story for this week. It's a short one. I hope you found this interesting. I have no idea what we're going to talk about next week. Sign up for the channel, like it under the comments, sign up for alerts or not. I don't care so much. Thank you for watching. I wish you all a good weekend. I'm going to call Currys now to ask them to take away this new washing machine. And I hope to see you next week. Bye-bye.